Welcome to today's episode of Molecules to Medicine podcast. For those of you just joining the podcast, we are AI Jules and we specialize in helping healthcare and life science companies scale their business and revenue and explore how AI is reshaping the industries and accelerating innovation. So, if you'd like to learn more on how to take your business to the next level, make sure you stick around. And if you're interested in a free growth consultation, just drop us the word growth in the DMs and we'll get back to you. I am your host, Hadi, and we have a very exciting guest for you today, Michael Liebman, the managing director and co-founder from IPQ Analytics. Welcome, Michael.
Welcome. Thank you. Thank you
And
for
where are
the
you?
opportunity.
No problem. And where are you talking to us from today?
Just outside Philadelphia in the U.S.
Fantastic. So, Michael, tell us a bit about your background and the journey that led you to IPQ Analytics and any pivotal moments or experiences that shaped your career.
Sure. So I started out as a theoretical chemist and protein crystallographer. I had an academic career, went into industry, eventually landed as global head of computational biology and genomics for Roche Pharmaceuticals, and then went back into academics at the University of Pennsylvania and was recruited to run a breast cancer center for the U.S. Department of Defense jointly with Walter Reed, where they did everything from surgery, tissue banking, genomics, proteomics, and I created a personalized health record that they've been using for collecting data. From that, we launched eventually IPQ Analytics. It's a modeling as a service organization. We work internationally building models of disease, but from the clinic back rather than from molecular processes forward. And we use that to stratify disease, improve diagnosis, and so on. We work directly with pharma and biotech, healthcare providers, payers, and government agencies, again, as I said, internationally. And then this past year, we've launched a nonprofit called Woven that's focused just on women's health. And so that's been a large part of our activity this past year.
Okay, fantastic. And tell me a bit about IPQ Analytics and what is it specifically that you do and who do you do it for?
Well, one thing when I was running the breast cancer center that I became aware of in working with both breast surgeons and oncologists and pathologists was, to be honest with you, some of the limited understanding of the disease. And I do systems modeling. And so the concerns I had were about quality of diagnosis. What are the gaps in understanding the process of diagnosis, which of course impacts treatment, also impacts the development of drugs. If you don't have the right diagnosis, you don't have the right targets to look at. And so as I started to build models, what we were doing is looking at how to stratify the disease. So almost all diseases are complex, and that means that there are a number of different subtypes. A lot of the effort today focuses on driving an understanding of the subtypes from genomics. We're interested in driving it from the actual presentation of the disease back and then seeing where genomics may tie into that, but not purely driven by genomics. So that means we work with physicians and we work with patients quite a bit to understand their experience and really to understand how they go about making a diagnosis. It's very easy to analyze the data that you have. I mean, in principle, it is, but that doesn't necessarily mean you have the right data to start with. And that's a lot of what we focus on. So in this world today, almost everybody asks me, are you an AI shop? And do you do machine learning on big data? And the answer I always give them is no, we're not an AI shop. We're a problem solving shop. And what we're interested in is the reason we don't purely do machine learning on big data, which has value because you're able to find patterns in big data that you may not have observed, is that that big data was generated because someone had a model and that data reflects that model. And therefore, you're analyzing that model without an ability to go beyond that model. So rather than doing data-driven modeling, we do what I call model-driven data. We build a model based on fundamental concepts and then look to see if the data is available. If the data is not available, we can identify gaps and conflicts and actually resolve a number of conflicts that way. In some instances right now, we're collecting data internationally to fill some of those gaps or understand what the limitation is of the data that's available. So we are using an AI component because we've developed a novel way to represent knowledge graphs and be able to do analytics off the knowledge graphs that support that model-driven data approach. So that's sort of a summary. I've worked in about 20 different diseases, and our women's health focus in Woven, as I explained to people, we're not doing femtech, we're doing femhealth, which is a very different topic.
You mentioned there you worked at Roche and you're working with women's health and breast cancer. Were you involved in Herceptin and HER2 testing um, for diagnosing breast cancer?
I actually was involved in the original HER2 fish test development,
Oh.
and part of my job was to figure out what to do with the test because Herceptin wasn't out yet. And so I built models of how the HER2 fish test could be used to change the practice of medicine, even if we didn't have a drug yet. So yes, I've been involved in that for quite some time, and that also triggered a lot of my interest in learning and recognizing that we really didn't understand breast cancer because we're very much not interested, not focused on correlation that you get from machine learning. We're interested in driving causality because we think that's the key to really making a difference in disease management or drug development.
It's interesting you say that because in my earlier days of my career, I worked in the lab where we did a lot of the HER2 testing on tissue samples
Okay.
to see which patients would be susceptible to Herceptin. So in the histology labs, that is. And using the fluorescent in situ hybridization method as well on the machine. So I guess that would have been around that time you were working on it.
Yeah, I was working on it back in the mid 90s. Exactly.
Yeah, yeah. This was more towards early 2000s, mid 2000s, where it was actually probably now
Yeah.
implemented
Yeah.
across healthcare. Oh, fantastic! And what problem does IPQ try to solve, and why does it matter to your customers?
Well, what I found when I was in pharma and also found when I was in academics is that there's a lot of good research that goes on. In pharma, I spent 11 years in pharma, and the challenge that I found, and it started when I was working on HER2, was the lack of understanding of the disease as opposed to trying to use some of the advanced technology to identify a target that would be amenable to drug development. And what I was always trying to convince pharma about was if we had a better understanding of the disease, we come up with a much earlier and better definition of the target rather than find a target, develop a drug, and then see if it works in humans or animals. So that was a frustration I had in pharma. When I went back to academics after being away for 11 years, the frustration was there was excellent research being done, but almost none of the research was focused on what was the clinical need. It was focused on what was a good research problem. And so when I left, it was really to try to bridge that gap. How do we actually bring that gap to be a very narrow and accessible gap? At one point, I was involved with PHRMA, the organization, and involved in the start of their translational medicine program. And what I defined when we did that, whereas everyone was defining translational medicine as moving things from the lab into the clinic, my approach was you start in the clinic, not in the lab, to bring the problems to the lab because then you will be able to translate solutions much more effectively. And so we've really looked at that bi-directional translation as being critical. And that's been the biggest challenge. It's not just identifying the clinical problem, but also working with clinicians, practitioners, and patients to understand what they are because they're not always used to thinking about what's the real root cause problem I have because they have to be very operational. And so that's part of the challenge is getting to those unknown issues.
And are you finding there's a shift in thinking to adopt this? What sounds to me a logical rationale.
Well, it's interesting. I was just at a breast cancer conference and there were excellent clinicians, and so on, oncologists. And what you see is, I guess, a little bit of what I call reduction to the complexity of the problem. So there was a lot of discussion about prevention, but what I saw was not prevention of causality. It was prevention of early detection. So how do you get early detection to prevent it from progressing? Not how do you prevent it from happening to start with? And it's that kind of challenge that you run into because if you talk to practitioners one on one, they admit to what the challenge is, but it's not what they're trained to actually do because they have to be operational and deal with patients. And that's what I've tried to build is a relationship with them to support their being operational where we can be more strategic. We can take that longer view and ferret out what those really complex questions are, such as, you make a diagnosis, but maybe that disease has 5 subtypes. For reimbursement purposes, you have to give that ICD-10 code, but that ICD-10 code doesn't describe the real biology, and that doesn't necessarily tell you the optimal treatment at a personalized level. And that's the kind of thing we're trying to drive to. So we're very focused on stratifying the disease, coming up with more selective diagnostic criteria, being able to use that to better treat the patient, as well as to even better design drugs or develop policies, or even for payers knowing what you should or shouldn't reimburse.
Okay, fantastic. So, and are you using any sort of softwares to find these disease targets? How exactly are you modeling it to find these?
Well, what we've done is we've taken the knowledge graph approach, which for those in computer sciences is a form of object-oriented programming from years ago, because you're looking at a very broad set of concepts and relationships and expanded upon that. Because currently, knowledge graphs are being used to represent existing knowledge, papers and so on, extracting from them the concepts and relationships and building a graph. And then you can run, agents across that to find complex interactions. But that approach focuses on what I call known knowns and known unknowns. We're interested in the unknown unknowns. And so what we've done is rather than use the knowledge graph as it's developed now, we've expanded that in what we call expanded known unknowns and determine what are the parameters that really should be examined that we're not examining that may be critical. So, I'll give you 2 examples that might be helpful. At one time, we were working in kidney transplant, and one of the parameters that's used on transplant guidelines is glomerular filtration rate. Now, to measure glomerular filtration rate, you should put the patient in the hospital and look at their total input and output of fluid, because that's what it is. But we don't do that because it's not convenient, so we use estimated glomerular filtration rate. And so every health record has your EGFR, which is computed based off of ratios of lab parameters. What it doesn't tell you is that there's 5 different equations to actually compute that because they were developed for different reasons, and it doesn't tell you which equation was used, and they don't give you the same numbers. Now, that doesn't mean that that's the most critical number in determining something, but it gives you an idea of the lack of adequate context that's present in medical records that could have an impact, especially if it's compounded with other things. So that's one of the kinds of things that we see that's very important. We also, when we're looking at, say, risk algorithms, and we talk about family history, we talk about the example I use is parents with 4 children and say that they have an equal sharing of genetic risk. And that's what the textbooks look like. But the real world is that those 4 children weren't born at the same time and that those parents were different before each of those pregnancies. And so there's epigenetic differences that could be very significant, but we don't incorporate them because it makes it too complex.
Uh-huh.
And those are the kinds of things we go deeply into to make transparent, not to try to say you can't address the problem without all that data, but you do need to know what you aren't considering because it may be important, especially when your problem isn't being solved by the approach you're taking.
Going back to your work with kidney disease and the EGFR numbers there. So the work you do, I mean, how would that translate to a patient outcome? I mean, can you sort of exclude the need for a biopsy, for example, a renal biopsy using the numbers, something like that?
No, but I'll give you the perspective. I spoke to a kidney transplant specialist at a very highly respected institution that I won't name for obvious reasons. And I pointed out to him this, and I asked him what they did about it. And he said, it's true, it's real. We do get differences in those numbers. I said, so what do you do? Because it's in the guidelines that you have to evaluate this. And he said, we ignore it. We don't use it. And so what you're seeing is the guidelines are not necessarily what are being followed for real world practice, which is acceptable because they're guidelines. They're not mandates, and a physician has that liberty, but it also should point you to where we need to improve how we're developing guidelines by recognizing that complexity, even designing experiments where we test that out to see what would happen with those different values. So I can't tell you that it's going to remove a biopsy opportunity, but I can tell you where we would try to apply it and where it may show up that'd be significant.
Okay, fantastic. And what's the biggest challenge or bottlenecks you face in getting yourselves with the right type of client?
Well, there's two challenges. One is what we're doing is we're treating disease as a process that evolves over time. So it's not what you look at as a disease state. So we define it mathematically as a vector that moves in high dimensional space where those dimensions are all the clinical parameters that could be measured. How far along that vector you have is that's what a stage of the disease is. And then disease has a velocity, and it may not be constant at all those points. So to really define disease, we should be defining it as a tensor, which we don't have the data to support. So the challenge is that two patients, when they come in, may look identical, but not actually be identical just because of when we're sampling them in their disease. Or two patients may look very different, but have actually the same disease, but again, because they came in at different points in their disease, look very different. Then you add on top of that the fact that the average patient has up to 5 comorbid conditions, which are going to alter a lot of those trajectory issues. And now you have the complexity of how do you make a diagnosis? And what you need is longitudinal data. US is not very good at collecting longitudinal data. There are only a couple of places where that data actually is collected. And we're trying to work with both of them because we want to track an individual's progression from well before they're ever diagnosed to understand what the pivot is at that point and what path they may be on. In our women's health program, Woven, the nonprofit, one of the things we're looking at is the fact that in perimenopause, we treat women as if they're pre or post menopause. But the reality is women are progressing on different tracks through perimenopause. We're not managing them effectively because we don't have data to define that. And what we're seeing is those tracks begin as early as puberty, but not actually show effects until much later on. We need to understand that that data can be critical, even though it's not currently being collected, because if they're on different tracks, they're going to have different risk for breast cancer or cardiovascular disease or osteoporosis, along with their symptoms they present in that 7 to 10 year transition period. So since the data doesn't exist, one of the challenges we have is we're trying to collect the data. And that's the kind of thing that we get involved in. Can we collect the data? And if we can't, can we simulate enough of it to understand what the risk is of not having the data? Not to add simulated data, but rather to evaluate what the limitations are and what we can quantitatively state is accurate without that data.
Fantastic. And what makes IPQ Analytics different from others trying to solve the same problem?
We don't have people who are really focused on looking at stratifying the disease the way we're doing it, because we're doing it on symptomology and we're doing it on clinical presentation. Most of the stratification that takes place right now is based on genomics. And it's a start, but it's a component, but it's not necessarily adequate to really define the disease because you need to look at a lot of other factors: clinical history, environmental exposure, lifestyle, social determinants of health, which we've expanded to also look at cultural determinants of health and trust determinants of health. And as I pointed out earlier, it's some of the epigenetic differences that may be very critical. So we're not relying on a technology to try to stratify the conditions, which would make it a lot easier if we had that, but to try to actually look at a trajectory over, in this case, a woman's lifetime. And we're doing that with other diseases. We're doing that with multiple sclerosis. We're not looking at the issue at time of diagnosis because we know in the process, it takes about 5 years to come up with a diagnosis by ruling everything else out rather than ruling MS in. And that means that that patient, when they are getting their diagnosis, probably of relapsing remitting MS, has also probably been treated with drugs to manage the symptoms. And so part of the question that should be asked is, what disease do they actually have at that point? Because it's been modified by the drugs and the disease progression. So we're going all the way back to interview these patients to find out, potentially in a prodrome approach, what symptoms might they have had very early on that maybe they didn't even go to the clinician to report because they were very transitory. It could have been dizziness, it could have been fatigue, it could have been muscles, and so on. And we're actually trying to collect and analyze that data to see how early we may have detected the potential that someone's at risk. So those are the kinds of things that we're doing differently that a lot of the other groups have not yet taken on as a challenge. But that's also why we're doing it in the nonprofit, because pharma wasn't in a position to do that. And in academics, they're not necessarily in a position to do that. In fact, after a presentation I made at a breast cancer conference, a deputy director from NCI approached me and told me that this was really the way we should be doing the science. And it was really good to see that we're doing it, but I would never get funded at NIH to do it. So it's not the way science is being done, and we're just trying to acknowledge and deal with the complexity, not make it something that has to be completely addressed, but make it more transparent. So when things don't work, we may know why.
Fantastic. And where do you see the biggest opportunity for innovation right now, and what excites you most about the future?
Well, we see that this can be applied a lot of different ways. Ideally, we would think it should be applied in discovery, but discovery is very noisy, having been in there, because everyone wants to get into discovery, but discovery actually doesn't recognize its value for many years post the activity. So we've been more directed at interacting with clinical trials, and in particular, trials that failed. Because the question is, why did they fail? And we all know that these trials have been dealing with patient populations that are not real world patients because they're selected to minimize additional comorbid conditions, etc. So when a trial fails and subgroup analysis is being applied, it's further moving that patient group away from the real world. What we're doing is we're working with the clinicians in the real world data, not claims data, because obviously that's tied to reimbursement, but real world data to build these stratifications of the disease and then actually go back into the trial with new diagnostic criteria and re-diagnose the patients to see how that drug may have actually worked. And that's an opportunity then to really not reposition the drug, but refocus the drugs on their original indication, but now with a better resolution on who the patients are that would be most susceptible and respond to those drugs for those conditions. And that's one of the things, because it recovers value in this, but it also means that there are drugs, a lot of drugs probably right now, that because they failed the trial for a variety of reasons, may not actually get to patients. And so the patients aren't getting benefit, even if there might only be a small segment of patients, because we're not adequately resolving how to identify those patients.
And is there any way that you could facilitate maybe any reformulation that a drug may need because it's failed a trial or
Well,
produced?
yeah, exactly. That's what we're doing.
Yeah. Okay, so it's actually helping going back to the lab and tweaking the drug for any
Yeah.
products. Yeah. Okay, great. Brilliant. And what stage is IPQ Analytics at the moment as a business, and what does growth look like for you in the future?
Well, IPQ Analytics has been focused on a modeling as a service consulting opportunity that works internationally. And we work with a lot of underserved populations. It's all been privately held up until now. And again, we've just launched the nonprofit this past year. Again, that's currently being funded through IPQ. We are looking at expanding that activity. I mentioned that we're working in perimenopause, menopause. We're also working in infant and maternal morbidity and mortality. So we're looking at things like hypertensive disorders of pregnancy. We're doing some feasibility studies, because what we'd like to do is be able to launch some longitudinal studies and attract some investment based on the results of the feasibility studies to show the value of initiating a really long longitudinal study starting at least at puberty, if not earlier.
Okay, great, fantastic. And where can people learn more about IPQ Analytics or get in touch with you or your team?
Well, they can reach me directly at michael.liebman, Liebman, at ipqanalytics.com. They can go to www.ipqanalytics.com, and there's women's health section there that describes our nonprofit, and that will soon be launched as a separate website as well. It's called Woven.
Thank you very much, Michael. Very, very interesting. And I wish you all the best for the future for yourself and the business.
Thank you.